Okay, now that we've learned how cells take apart food molecules, we're going to learn about how other cells that have chloroplasts, are going to be the ones that trap the solar energy and use that energy to rearrange the atoms in carbon dioxide and water and make food molecules. So just as you broke down glucose and you have the products of ATP, water, and carbon dioxide, we're going to be taking water and carbon dioxide and the energy from the sun and assembling these molecules. So it's in a lot of ways it's the reverse of what happens in cellular respiration. Now, solar radiation obviously has a bunch of points of significance in our lives. One of the reasons why you do things like wear sunscreen is that short wavelengths of light are more powerful. They have more energy, and so short wavelengths of light can do things like pass into the tissues of your body, like passing into your skin. The very long wavelengths of energy are much weaker. So things like a radio signal, they're much longer and they're much weaker. So something like short wavelengths, the shorter the wavelengths, the more energy there is. So something like X-rays can go right through you literally, but the stuff you meet outside in the sunlight, some of those shorter wavelengths can damage the genetic material in your skin and lead to things like tumors and cancers and whatnot. Okay. And I mentioned the other day about carbon sinks, the fact that plants take in carbon and use that to assemble everything from the cellulose that makes up their cell walls to nutrient molecules like glucose. So one thing that you can do to combat climate change is plant trees. And sometimes you'll see something like a company will be, for every X you buy, we plant trees. And so it's kind of interesting. There's all these different ways in which people are trying to increase the amount of carbon sinks, right? Things that will absorb carbon from the atmosphere and tie it up in organic tissue. And then that's so much less carbon that's, pardon me, out there to increase the planet's warming. And as I said before in the previous section, nearly all life on Earth, including us, can trace our sources of energy back to the sun. The exceptions from the last section were things like deep sea vent bacteria, but we don't live down there. So pretty much up here, this part of the biosphere, it all really traces back to sunlight. Now, something that some of you may have or at least have seen are solar panels, right? You have these solar collectors, these flat panels that absorb sunlight and convert that solar energy to electricity. It can be used right away, it can be stored in batteries, or people who generate enough of it can actually transfer it, push it backwards into the electrical grid. And in some places, they have to pay you for that. So if you have a really good solar setup, you can actually not just save money, but eventually even make money. Now, living things that capture sunlight using photosynthesis can also serve as energy sources. Right? Plant matter can be burned. If you think of burning wood or even coal, coal is just very ancient plant matter. Or you can use plant materials to produce biofuels. And that's another area of renewable energy. Stuff like coal is non-renewable energy. You're not making more coal. That's millions of years old dead plants. So if you can find ways of making fuel out of plants that you can keep growing, that gives you a bit more flexibility and something that's a bit more renewable. So there's various ways of capturing the energy of sunlight, either solar panels, burning plant matter, or growing things that can be used to produce fuels. And again, you can see pretty routinely now houses with solar panels on them. Now up here, in the northern zones, it's probably more of a supplement than a complete source of electricity, though a small efficient house probably could do it. But again, if you have solar panels, you're saving money, you're causing less burning of fossil fuels, so not much of a downside. Okay, photosynthesis. Photo, light, synthesis, create. So synthesizing organic molecules using light as an energy source. So we typically think of plants, the big eukaryotic organisms, but there are also algae, which are actually a type of protist. Even some of the bacteria, the blue-green bacteria, all transform light energy into chemical energy. The starting materials are inorganic, carbon dioxide and water, and the byproduct is oxygen gas. So again, one organism's waste product is another organism's necessary reactant. So this chemical energy produced by photosynthesis, using sunlight energy to rearrange these atoms in carbon dioxide and water, that chemical energy is then stored in the bonds of sugar molecules. So sticking together those carbons and oxygens in a certain pattern, that's a way of storing energy, right? So that sunlight energy can get trapped and converted into a chemical form. So any organism that generates their own organic matter from inorganic ingredients are called autotrophs, literally self-feeding. So plants that do this by photosynthesis are formally called photoautotrophs, and those are the producers for most ecosystems. Down in the deep sea, you had those chemoautotrophs, but again, we don't talk about that so much. We usually are, when we say autotroph, we're usually referring to a photoautotroph. And again, the fact that nearly everything, pretty much everything on the surface of the Earth can trace their source of energy back to the sun illustrates that the ability to transform energy and matter is vital, right? You're not going to have life on Earth without the ability to transform energy into usable forms, transform matter into usable forms. And I just wanted to show you, some examples of photoautotrophs. Again, what most of us think of are terrestrial plants. But if you've ever seen seaweed, otherwise known as algae, these are a type of protist. They may look like a plant, but they are not a plant. They're anatomically, they're very different. But functionally, big flat surfaces with chloroplasts. So in a functional sense, they resemble plants, but they are not. It's a whole different group, and we'll get to that later in the semester. And from our last section, here's some of those cyanobacteria. So you don't have to stain these. They actually look, cyano, referring to a blue-green color. So the organelle chloroplasts, they're light-absorbing organelles. They are the site where photosynthesis takes place, and they're found mostly in the interior cells of leaves. That green color comes from chlorophyll. So whenever you see phyll, that's usually referring to a pigment. So chlorophyll is green pigment. So this pigment, and the definition of a pigment is anything that can absorb light. So chlorophyll is a pigment, a light-absorbing molecule, located in the chloroplasts. Chloro, again, green. Plast usually refers to a small structure in a plant cell. Like chloroplast, starch structures are amyloplast, right? So there's various little bodies in plant cells. So chloroplasts are these specific organelles that are carrying out the converting of solar energy into chemical energy. Now, one of the things you needed was carbon dioxide, and one of the things that's given off is oxygen. So how do you do gas exchange? There's things called stomata. The word literally means little mouth. And they're little pores in the leaves where carbon dioxide enters and oxygen exits. Now there's membranes inside the chloroplast which form the structures where the reactions of photosynthesis occurs. So just as you had membranes in the mitochondria, which had those proteins which carried out the electron transport chain, you have membranes in the chloroplast where we're going to have certain reactions taking place. So again, there's a lot of parallels between the two. So let's sort of dive into a leaf. So if you took a slice of a leaf and looked at it in cross-section, so right, the first photograph, you're looking down onto the leaf. But the image you see next is actually a slice taken out of that, and you're looking at the cut edge of the leaf. And up at the top, you see sort of vertical cells, right? They're sort of like little parallel, columns. Those are the cells on the top surface of the leaf, and they're packed close together and they're loaded with chloroplasts. Well, because just like a solar panel, you want to absorb as much sunlight as possible. But when you look on the underside, you see that they're sort of disorganized. They're also cells that have chloroplasts, but they're sort of messier, right? It looks almost like a sponge with little holes and spaces. That's because, well, whereas that top layer is doing a lot of the sun absorption, that bottom layer, you'll see at the very bottom surface, I don't know if the pointer will work in this context. But you can see what looks like a little pair of lips down here in the bottom layer of the plant. Those are the stomata. The reason why these are all, spaced around and there's gaps between them is that this is where the gas exchange is going on. So if they were all packed closely together like the upper layer, it would be hard for gases to move around those cells. Here, there are spaces around the cells, so gas exchange can happen much more easily. So that's why anatomically, you actually see the cells arranged differently depending on whether their primary function is absorbing sunlight or carrying out gas exchange. So if we zoom into one of those little tiny green dots in one of those cells, okay, so say we're looking at an interior cell here, like one of the guys at the bottom side of the leaf, we zoom in on one of those organelles, and that organelle is a chloroplast. Now, the outer membranes sort of package the whole thing. The inner membranes, instead of looking sort of like waves in the, as you saw in the mitochondria, they look almost like little stacks of pancakes. So these little structures, okay, they sort of zoom into one of those stacks. So the stacks are called granum. Each individual disc is referred to as a thylakoid. And you can see a little stack of these guys. So each of these little stacks of granum contain multiple folded membranes, which is where these reactions are going to be taking place. So again, if you have a reaction taking place on a surface, you want to increase the surface area to make it more efficient. So just popping in a little closer here. So again, you can see the upper layer of photosynthetic cells packed close together, and then the lower layer more loosely arranged. And again, that's because gas exchange is happening on the underside, whereas it's primarily light absorption going on the upper side. Now, obviously, they exchange materials with each other, but there is some regionalization of how they do their work. And again, somewhat larger illustration, and let me pop to that last image there. And here you can really clearly see those thylakoid discs forming the granum stacks. So again, all these tiny layers you see here, these are all membranes where you're going to have reactions taking place. So, like the mitochondrion, the chloroplast has a double membrane envelope. The inner membrane encloses a compartment filled with stroma, a thick fluid. Okay, so you had a thick fluid inside the mitochondrion, which is the you just lost the word, matrix. There's a fluid inside the compartment of the mitochondria. There's a fluid inside the compartment of the chloroplast, and this compartment is called stroma, so that's the fluid. Suspended in that fluid are the interconnected membranous sacs called thylakoids. And then the thylakoids are concentrated in stacks called grana, and the chlorophyll molecules that capture the light energy are built into those membranes. So obviously, if you've got something important taking place in a membrane, more membrane means you can get more done. Now, if you look at the overall equation for photosynthesis, you're basically looking at the reverse of what we saw last time in cellular respiration. The reactants of photosynthesis, right, the things that go into the reaction, are carbon dioxide and water. Those are the same as the waste products of cellular respiration. And what does photosynthesis produce? Glucose and oxygen. That's what respiration uses. Those are the reactants going into respiration. So, like that diagram from the other day, we're basically just looking at the reverse of it, right? 6 oxygen molecules and glucose being constructed from 6 molecules of carbon dioxide and 6 molecules of water. The big difference being that instead of taking it apart to yield energy, you're using energy to put it together. Now, the overall process of photosynthesis can get broken down into 2 stages, connected by energy-carrying and electron-carrying molecules, right? So just as we had electron carriers in cellular respiration, you have electron carriers in photosynthesis. And you actually use ATP in the process of making glucose. So you actually see also the same energy-carrying molecules. Now, the 2 reactions are called the light reactions and the Calvin cycle, sometimes also called the light independent or dark reactions. And what you can get from those names is that one needs light to take place and the other does not. So the light reactions are the ones where chlorophyll in the thylakoid membranes absorb that solar energy, which then gets converted to the chemical energy of ATP and NADPH, right? These are characters we met last week, or in cellular respiration. The second stage is the Calvin cycle, and these use the products of the light cycle, the light reactions rather, to power the production of sugar from carbon dioxide. So, sort of a basic roadmap. So here's your light reactions. So, water, which is taken up in the case of a vascular plant or regular land plant. Water is brought in from the roots, right, and it travels through those little veins out to the leaves. Okay, gas exchange is occurring through those stomata, the little openings. So you get the water absorbed. The light reactions are going to do is cause energy changes that are going to split water. All right, so you're going to take that water and split it. You're going to load up your electron carriers, and then the oxygen is let go as a waste product. In these other reactions, okay, this is where the carbon dioxide comes in. The carbon dioxide is going to be the carbon source, right? We talk about glucose being that 6-carbon molecule. Well, where's the carbon come from? Carbon dioxide. So just as in respiration, the carbons from glucose were given off as carbon dioxide, here the carbon dioxide is being taken up and being used as the source of the carbons for the sugar. So again, there's a lot that's sort of a mirror of what we saw. So you have the loaded electron carriers in ATP bringing energy to the Calvin cycle, and they're going to be used, and so the ready to be reloaded NADP plus and the NADP plus P go back to the light reactions, right? So just as you had these electron carriers and light and energy carrying molecules in cellular respiration, we see a lot of the same players here in photosynthesis. So that initial incorporation of carbon from the atmosphere into organic compounds is called carbon fixation. Whenever you take a gas and trap it into organic compounds, we refer to it as fixation. There's nitrogen fixation. In this case, it's carbon fixation. And again, this is one way of reducing the amount of carbon dioxide in the atmosphere because you're binding up that gas into organic compounds. So that relationship between photosynthesis, carbon fixation, and global climate is a really great example of how interactions between biological components at many different levels affect everything on Earth. So here's the video.
Plants provide us with food to eat and oxygen to breathe. They perform this amazing feat by the process of photosynthesis. Let's take a closer look. Photosynthesis requires carbon dioxide, which diffuses into the leaf through small pores and then enters the cells. Inside the cell, carbon dioxide diffuses into the chloroplast, where photosynthesis takes place. Chloroplasts use energy from light to transform carbon dioxide and water into sugar and oxygen. Zooming into a chloroplast, we see these flattened membranous sacs called thylakoids. Here, light energy is converted to chemical energy in the first phase of photosynthesis, the light reactions. The two green structures you see here are photosystems, large complexes of proteins and chlorophyll that capture light energy. An electron transport chain connects the two photosystems. Notice the small mobile electron carriers that shuttle electrons from one large complex to another. Now let's take a closer look at the steps of the light reactions. The photosystem on the left absorbs light energy, exciting electrons that enter the electron transport chain. Electrons are replaced with electrons stripped from water, creating oxygen as a byproduct. The energized electrons flow down the electron transport chain, releasing energy that is used to pump hydrogen ions, the blue balls, into the thylakoid. In the photosystem on the right, light energy excites electrons, and this time the electrons are captured by an electron carrier molecule, NADPH. The high concentration of hydrogen ions inside the thylakoid powers ATP synthase, producing ATPs. The light reactions in the thylakoid have produced two energy products, ATP and NADPH, that will now power the production of sugar in the Calvin cycle. The Calvin cycle takes place outside the thylakoids in the stroma, the thick fluid of the chloroplast. At the beginning of the cycle, carbon dioxide molecules combine with molecules called RUBP. The resulting molecules go through a series of reactions powered by ATP and NADPH from the light reactions. Sugar molecules known as G3Ps are produced. Most of the G3Ps are rearranged back into RUBPs that will begin the Calvin cycle again. But the important product of photosynthesis is the remaining G3P sugar. Some G3Ps are used to build glucose, which can combine into starch or cellulose. Still other G3Ps form sucrose. And some of the sugar is broken down by cellular respiration using oxygen in the plant's own mitochondria, generating ATPs that can power other work of the plant. Excess oxygen diffuses out of the leaf through the pores, while more carbon dioxide enters. With three simple ingredients, carbon dioxide, water, and light, plants produce sugar and oxygen by photosynthesis, powering plant metabolism, and ultimately providing your fuel as well.
Okay, so that sort of gives you a nice synopsis of the whole thing. So let's go through these in steps. So starting with the light reactions. This is the point where you're converting solar energy to chemical energy. So let's talk a little bit about radiation first. I mentioned it at the very beginning of the chapter, but I want to go into this in more detail. Sunlight, like other forms of radiation, travels through space as rhythmic waves, right? So you think of waves of water where you have a crest and a trough, right? A high point and a low point. The distance between the crests of two adjacent waves is called a wavelength, right? That's how long the wave is. The full range of radiation from the shortest waves to the longest waves is called the electromagnetic spectrum. So if you look at this diagram from left to right, you're looking at increasing wavelengths, right? So things like gamma rays and X-rays are really, really short. Things like radio waves are very, very long, right? And that number at the top of the bar tells you how long the wavelength is. So whereas something like you talk about things like microwaves, that's a good example. You're talking about wavelengths up to a meter, right? So like 3 feet between crest to crest. Whereas if you're talking about some of these smaller wavelengths, right, you're getting into things where you're getting into nanometers and fractions of a nanometer, right? So there's a big range of wavelengths. So if you start looking at that gray bar, start at the left, gamma rays and X-rays, these are short ones, right? If you wanted to do something like take an image inside a person, you'd use something like an X-ray, right? That would penetrate all the way through, and the radiation would be absorbed by things like hard tissue. Something like UV, that's a bit longer, right? So it doesn't drill all the way through you like an X-ray, but, it can penetrate your skin. The next part, you see a little gap in there. That little space there is what we call visible light. In other words, visible to humans light. All right, that little tiny space there, if you look at if you think of everything you can see around you in terms of colors and all that, that's just that tiny little bit in that little gap there. Now there's organisms that can actually see further, beyond that range. So there's a lot of insects, even birds, reptiles, that can actually see UV. There's actually patterns on some flowers that we don't see, but bees can see. I knew somebody who studied a type of lizard, and people wondered why they made a little waxy material on their thighs. And they'd see them rub it on a rock, but it's like, okay, didn't know what was going on until they started shining UV light on it, and it turns out that, they're marking these areas in ways that other lizards can see, but nothing else like a predator would be able to see. So we're sort of learning that it's way beyond us. If you go over to the infrared side, right, so this is now wavelengths that are longer than the red that we can see in the 700 to 750 nanometer range. There are things like rattlesnakes have a little, what looks like a little pit between their nostril and their eye. They can actually detect infrared radiation. So in the dark, they can detect something that's warm from that, slightly longer than red wavelength radiation. So it's kind of neat. We always think of stuff being us, but there's perception beyond our range. So jumping down to that visible light bar, okay, you see it goes from violet to indigo to blue to green to yellow to orange to red. I'm laughing because I learned it as Roy G. Biv. Red, orange, yellow, green, blue, indigo, violet. So it's hard for me to say it backwards. So you're going from shorter wavelengths down at the purple to blue range to longer wavelengths, and you're getting into the orange to red zone. And again, at the bottom, they give you an example of a wavelength. So that space from wave to wave is the wavelength. So when you're looking at that 1 point of orange, the wavelength's about 580 nanometers. And finishing off that gray bar, so again, progressively longer waves are microwaves and radio waves. So again, the longer the wavelength, the less the energy. So how did people figure out which wavelengths are involved with photosynthesis? Well, in the late 1800s, Theodor Engelmann saw that certain bacteria living in water, because remember there's those blue-green bacteria, they cluster in areas with higher oxygen concentrations. It's like, So I'm sorry, the bacteria that gathered were not the actual photosynthetic ones. My apologies. Certain bacteria living in water cluster in areas with higher oxygen concentrations. So these are things that need to take up oxygen. So if there's more oxygen, these guys are going to pile up where the oxygen is. So he formed a hypothesis that oxygen-seeking bacteria will gather near regions of algae performing the most photosynthesis. So what he did was he took freshwater algal cells, and a lot of them are long strands. He took these algal cells in a drop of water on a microscope slide. And what he did was he said, okay, which wavelengths are going to be making the most oxygen? What he saw in the results, and I'll explain how he got there, was he saw most bacteria piled up around algae exposed to red-orange light and blue-violet light. And the way in which he split up the light was simply using a prism. Right? It's a piece of glass that will split light into its various wavelengths. So if you've ever taken a piece of glass and you see a rainbow forming, right, that's what a prism is doing. It's shining the color spectrum of visible light, and you can actually see the individual, wavelengths. So he did was he shone that rainbow, that spectrum of visible light, onto the slide containing the algae cells. He then added the bacteria, and then he looked on the slide to see where there were the most bacteria, and sure enough, you could see lots at both ends, lots at the blue end and lots at the red end, but not much in the middle, in the green end. And if you think about it, that makes sense, because when you see something that is green, the reason it appears green to your eyes is that that's the wavelengths being bounced off and striking your eyes, hitting the retinas, turning, firing up all those receptor cells. So it looks green because green is reflecting from it. So a selective absorption of light by leaves explains why they look green to us. Light of that color, light that is green, is poorly absorbed by chloroplasts. So it either bounces back or even passes right through the chloroplast, right? It's not being stopped. It's not being absorbed by the chloroplast. Whereas these other pigments are. So because that green escapes, it doesn't get absorbed, that's why we see the green. So, going back to section 1, energy can't be destroyed. So that absorbed energy, that energy that's absorbed by those pigments, have to get converted to other forms to be useful. So, again, back to our sort of an illustration showing, so if you look at light in terms of the spectrum, the absorbed light is stopped, right? So if you see that little light up at the top and it's an arrow going vertically through that chloroplast, right? All the wavelengths that are absorbed are not going to be escaping to hit your eyes, right? But green, because it's not absorbed, it's transmitted or bounced off, and those wavelengths will strike your eyes and will stimulate the receptors in your eyes, and so that's why they seem green.
The thylakoid membrane in chloroplasts contains light-absorbing molecules called pigments. Different pigments absorb light of different wavelengths. One pigment, chlorophyll A, absorbs mainly blue violet and red light. This molecule participates directly in the light reactions. Another molecule, chlorophyll B, absorbs mainly blue and orange light. We do not see these absorbed colors when we look at a leaf. Instead, we see the green wavelengths that are reflected back from the leaf or transmitted through it.
Now, chloroplasts contain several different pigments that absorb light of different wavelengths. Because again, pigment is something that absorbs, wavelengths of light, but the things that are different colors are absorbing different things. So chlorophyll A, as she said in the video, participates directly in the light reactions and is absorbing mainly blue, violet, and red light. Now, chlorophyll B does not participate directly in the light reactions, but it's kind of like the wingman. It's absorbing other wavelengths and then passing that energy along to chlorophyll A. So chlorophyll A is the one doing the work. Chlorophyll B is just sort of helping by bringing in more energy for chlorophyll A to use. Now, there's other pigments like carotenoids. So you see orange or yellow, which means blue-green light is what's being absorbed, right? If you're seeing it, that's not getting absorbed. It's bouncing off or passing through. So this time of year, up here in New England, you can see all these leaves turning orange and yellow and all that. One of the reasons that they are, quote, turning colors is that the chlorophyll's dying down. So the chlorophyll normally sort of covers up the other pigments. You don't see the other pigments because there's so much green. Once that green starts breaking down, now you can see the other pigments. And there's other metabolic stuff going on, but that's one of the reasons why you see those deciduous leaves changing in the fall. Now, one of the arguments that goes on forever with physicists is light a wave? That waving, energy, or is it little pieces sort of shooting along? And apparently they've decided to say it's both. That light behaves as a wave and as discrete packets, little pieces of energy called photons. So those are fixed quantities of light energy. So just as in, talking of light in terms of wavelengths, the shorter, the shorter the wavelengths of light, the greater the energy of any particular of little photons that would be associated with those wavelengths. The reason why I'm getting into this is that the pigment molecules, when we look down and closely at photosynthesis, we talk in terms of pigment molecules absorbing photons, so actual units of energy, instead of just one long flow of energy. So when a pigment molecule absorbs a photon, one of the electrons in the pigment, right, because pigments are just organic molecules, one of the pigment's electrons gains energy and is now said to be excited. So an excited electron is going to hold that energy briefly, but then it usually loses that energy and then goes back to its regular level of energy. So it gets briefly excited and then it loses that excess energy and it goes back to the ground state. Most pigments release heat energy as those light-excited electrons fall back to the ground state, right? So that energy, again, it's not being created or destroyed, so it absorbs it and then it gives it off. And most pigments release heat energy as those excited electrons return back down. Some pigments emit light as well as heat after absorbing photons. If you've ever seen a glow stick, you buy this little stick and you crack it and you mix two chemicals in there and you see a reaction take place. That fluorescent light emitted by a glow stick is caused by a chemical reaction that excites electrons of a fluorescent dye. So the electrons get excited, but as they fall back to their ground state, they're releasing energy in the form of fluorescent light. So instead of releasing it just as heat, it releases it in the form of wavelengths of light, which means you can see that glowing color. So fluorescing, referring to it appears to be glowing to our eyes. So let me go to this in a little more detail. So light, in the case of a chlorophyll molecule, okay, it comes in, hits the molecule, excites an electron in there, so it zooms up. So it's going to about what would be 12 o'clock on a clock face there. But it can't stay there. Right? It's only going to be there for a short while. So then it's going to go back to the ground state, but that energy that excited it has to go somewhere. So it's released in terms of heat and light. And if it's a wavelength of light that we refer to as fluorescent light, it can actually be visible to us. Hence the little glow sticks. So, in the thylakoid membrane, chlorophyll molecules, which are the guys doing the absorbing, are organized with other molecules into photosystems. Right, so if you think of a solar panel, parts of the solar panel are sort of holding things. There's other parts to it than just chlorophyll molecules. These photosystems, these organized groups, have a cluster of a few hundred pigment molecules, including chlorophylls A and B, and a few carotenoids. And that cluster together functions as a light-gathering antenna. So A is doing the major work, but B and the carotenoids sort of help by catching energy and handing it off to chlorophyll A. So here's an example of a photosystem. So again, zooming into the chloroplast, looking at those pigment molecules, embedded in the membrane. You then zoom into that, and I'm going to just go through these stepwise. So again, remember we talked about those pigment molecules embedded in the inner membrane. That's the thylakoid membrane. So we're going to zoom into that. We're going to look at it from the top. That's why it seems like a round image here. So what you have is these various molecules. And again, chlorophyll A is where the major reactions are taking place. The other pigments are basically there to catch energy and hand it off. So light energy comes in, right? It excites the electrons, and you're going to be doing is transferring excited electrons to the reaction center. And that's where the big work takes place. So let's go into this in a little more detail. I think it'll make a little more sense. So two photosystems cooperate in that light reaction. One, photons excite electrons in the chlorophyll of the first photosystem. Two, energized electrons from the first photosystem pass down an electron transport chain, where have we seen that before, to the second photosystem. The chloroplast uses the energy released by this electron fall to make ATP. Yes, just as we saw before. You're making a gradient, harvesting the energy of that gradient to make ATP. So that's just like we've already seen before. The second photosystem transfers its light-excited electrons to NAD+, reducing it to NADPH. So, here's the whole thing. Now, I will tell you something. When you look at this diagram, the one on the left is the first photosystem, the one on the right is the second photosystem. The reason why they say it that way is that the one on the right was discovered first. And if you look at books that are a little more formal, you'll see it called P system or photosystem 1. They call it that because they said, oh, we have the photosystem. Later on, someone discovered the one you see on the left side. So technically, it's photosystem 1 on the right, photosystem 2 on the left. That was the order in which they were discovered. Your book is making it a little bit easier on the brain by just saying the first one in the reaction versus the second one in the reaction. Just know if you're looking at a book that's a little more formal, you're going to see P system 1, P system 2, and it's going to seem like it's backwards. It's just because that's the order in which they were discovered. So your book is making it sequentially easier. Just don't be confused if you see it in another source, like a video online or something. Okay, so let's go in these in steps. So light comes in, right? You get that excited electronic, that you get an excited electron, and it's going to get captured by a primary electron acceptor. And energy that light energy is being used to split water. So you're going to get a source of electrons, a source of hydrogen protons, right? Because you're going to make that hydrogen proton gradient, and water, right? So just as oxygen was the last electron acceptor in cellular respiration, it's basically your first it's going to be your first electron involved in your first electron donor, right? The water is now donating electrons and giving off the oxygen instead of absorbing the electrons and binding with oxygen to make the water. So we're splitting water, getting electrons and protons, and dumping off the oxygen. So that primary electron acceptor, right? So you excited the electron with the solar energy. You split water with it. You got some electrons, you got some protons. But that excited electron is not going to stay there forever. So that energy is going to be trapped by a hydrogen acceptor, right? So it's going to do is instead of letting it just fall back, it's caught by the acceptor and then handed to the chain. So instead of just falling straight back down, it's going to fall down through a series of steps as it's getting passed along that chain, right? So instead of just boom, falling back down, it's now going to go in a series of steps. So it's still falling, but it's falling in a more controlled fashion. And so each of those steps, a little bit of energy is being used to push hydrogen protons across the across the membrane. And how do the protons get back? Through ATP synthase, right? So this is just like we saw at the electron transport chain. So you're producing energy. We're going to be using that later on. So just as the electron carriers used later on in cellular respiration, we're going to be using the ATP later on in this process. So those electrons get passed along the chain, just as you saw in the mitochondria, and they're going to arrive at that second photosystem, and more energy is going to, because the sunlight just keeps coming in, so sunlight's going to, excite that little guy again. So these are the first sets of reactions that require the light, right? So light's going to be coming in, exciting electrons, which can get grabbed by acceptors and passed along electron transport chains, right? So again, this is kind of similar to what we saw before. So the light reactions are located in the thylakoid membrane. The two photosystems and the electron transport chain are going to transfer electrons from water to NAD+, forming NADPH. So when you split that water, the oxygen left as a waste product, the hydrogen is being transferred with electrons to NAD+. This is sort of a slightly more detailed way of looking at it. So light just keeps pouring in, so it's going to hit pigments of both photosystems, right? It isn't like light's going to hit one and then it hits another. Light's just constantly hitting all these guys. So the thylakoid membrane, we start by looking at it in a sequence going from left to right here. So again, water is split, oxygen is released as a waste product, and those electrons are going to be sent off, right? These guys are getting excited, but they don't stay excited forever. So as they, instead of just falling back down like you saw the light energy in the glow stick, they're going to do is fall down in a controlled way. They're getting passed along the electron transport chain. And each step along the way, it loses a little energy. That's the energy being used to take those hydrogens from the water and pump those protons across the membrane. You make that big gradient, just as we saw back in cellular respiration. So what happens with those guys? They're going to pass through the ATP synthase. The energy is going to make ATP. In the second photosystem, there was electrons that passed, that got excited and were passing, basically moving, losing their energy downhill. When they land at that second photosystem, well, hey, more light's coming along. Boom, excites the poor little electron again. Except in this case, we're loading up, we're taking some of those hydrogen protons and loading up NAD+ to form NADPH. All right, so the electrons got used, all the way down to the second photosystem. They get excited again, but this time they get grabbed by the electron carriers. So 2 sources of energy are being formed, loaded electron carriers and ATP. Both of these are going to go off to the Calvin cycle. Right, so in the first photosystem, you get the excited electron. It's being passed along the electron transport chain, and that energy is pushing the hydrogen protons, which are going to run back through ATP synthase, making ATP. That spent little electron makes it, lands at the second photosystem and boom, gets hit with the light again, gets excited again, except now the energy is being trapped to form NADPH. So you're making energy in 2 ways. The ATP, from the products of the first photosystem, and then in NADPH when the light hits the second photosystem. Here's sort of a more fun way of looking at it. Right, so the guy with the mallet represents photons of light from the sun. So that energy excites the electron in that first photosystem, and it zooms up to the top. But instead of that person at the top just dropping it to the ground, she drops it onto a little paddle wheel. Right, that's the electron transport chain. So it goes downward in terms of energy more gradually, and that energy is being used to pump the hydrogen protons, create the gradient. The hydrogens then rush back through ATP synthase, and there's your ATP. But what happens to that electron at the end of the electron transport chain? Well, it lands at the second photosystem. More, because light's constantly hitting this, so another photon whacks it, and it gets excited again, but instead of going to an electron transport chain, it gets captured in NAD plus to make NADPH. Right, so those are the light reactions. Light is required to hit electrons in both of those photosystems to keep exciting these electrons, and then that energy that excited the electron is getting caught. Right, instead of it just being, the electron falling back down and the energy is lost as heat and maybe light, that excited electron, that energy from the sun exciting that electron, is being captured in molecules, captured in ATP, captured in NADPH. So that mechanism of ATP production during the light reactions is very much like what we saw in cellular respiration. Both cases, electron transport chain pumps hydrogen ions across a membrane, and ATP synthase uses the energy stored by that hydrogen proton gradient to make ATP. The main difference is that food provides the higher energy electrons in cellular respiration, whereas light excited electrons flow down the transport chain during photosynthesis. Now, those were the light reactions. Now we're going to go to the Calvin cycle, sometimes also nicknamed the dark reactions, because these don't need light to happen. In some plants, they actually capture a lot of the energy during the daylight and carry out these reactions when the sun goes down. Right? So these do not require sunlight to happen. Now, the Calvin cycle is where the actual sugar factory is taking place. It constructs an energy-rich sugar molecule called glyceraldehyde 3-phosphate, or G3P, using carbon from carbon dioxide, energy from ATP, and high energy electrons from NADPH. Think of the Calvin cycle, think of the Krebs cycle from the other day. Right? The Krebs cycle took this molecule, put it on a carrier, right? And as it went through the cycle, it got disassembled. You lost the carbons as carbon dioxide, you generated some ATP, and you loaded up some energy carriers. What's happening here? You're going to do is have a carrier move around a molecule, but it's getting built up using carbon dioxide instead of losing it. It's taking up energy from ATP instead of generating ATP. And it's getting electrons from NADPH as opposed to forming NADPH, as you saw in cellular respiration. So again, a lot that's very much just a mirror of the other. The big difference is G3P, some of it's going to be used to make glucose, right? So that sort of keeps the process going, like the reverse of cellular respiration. But some of it's also used as raw material for other organic compounds. So it isn't like all of this is going to be sugar. Some will be sugar, some is going to be making stuff like cellulose. So here's the Calvin cycle. So again, carbon dioxide coming in from the air instead of being released into the air as we saw in respiration. And that RUBP sugar is the carrier. All right, so that's the merry-go-round horse. So you're going to do is have carbon dioxide come in from the air. You're going to have a 3-carbon molecule formed. And again, you're going to have all these things going on with reactions along the way. The big thing is, again, the reverse of respiration. ATP is going to give up energy. This is energy that's going to be used to assemble this G3P sugar. And ADPH is going to give up energy. So these are being used to create this G3P sugar, which can go one of two ways. It can continue in the cycle, right, and keep going round and round, or some of it peels off and is used to make glucose or used to make other organic compounds. That 3GP sugar, some of them are going to do is go back to the cycle again. Right, so some of it serves as the merry-go-round horse, some of it leaps. So that's where it gets a little different from cellular respiration. In this case, some of the merry-go-round horses, the kids run off, and some of them the kids keep in the cycle. So that analogy breaks down a little bit. But keep in mind that it's a cycle. Some of those molecules keep going through the cycle again and again. Some get peeled off to be used for, as construction for glucose, etc. Now, we can say, okay, we want to take some of those other molecules and use them as a fuel source. But nature, the whole business of natural selection, the selective force was not making a biofuel. So we are trying to do is find ways of tweaking photosynthetic organisms to more efficiently generate, material that we can use as a fuel. So they're doing is saying, okay, just as you had that directed selection of an enzyme in the last section, they're going to use the same process to find the fittest, systems to make the most molecules that we could use for energy. So just as you generated a more efficient enzyme in the last section, here you're doing is, finding the organisms that are the most fit to make biofuel molecules. So this directed evolution of biofuel production often involves algae. Because if you think about it, you can grow unicellular or small chain algae in a solution, right? Like in a fish tank. Whereas plants, you've got to, you put them in the ground, you've got to grow them, if you have something that lives in just an aqueous solution, that's a lot easier to manipulate in the laboratory. So, and here's an example of someone working with these systems, and I think it was Exxon, I think, had a series of TV ads where they were showing, little flashes of researchers working on this. So similar to what we saw with the enzymes. We had, collecting algae. The algae get exposed to mutation-promoting chemicals. You then test these guys to see who produces the most useful biofuel in the largest, the most useful biofuel and the most useful in the largest quantity. The ones that do this best, okay, you save those guys. Grow a bunch of them. Subject those to mutation. Pick the ones that are efficient from those. Hang on just a sec. Repeated many times, this, is a way of efficiently producing biofuels.